Hello everyone, I would like to welcome you on QIAGEN's webinar on how can you characterize your CRISPR gene edits faster and easier, which will be presented today by Dr. Domenica Martorana. My name is Paula and I'm your host for today's webinar. Before we begin the presentation, we have a few housekeeping issues to go over. First, could you please find Ask Question box in your On24 panel and let me know whether audio is working and that you can hear me clearly. You can just type in hi or hello. All attendees will be listen-only mode and to avoid any background noises during the presentation. So if you experience any background issues and connection issues, such as you can't see the slides or you can't hear the audio, please type that in the chat box. In addition, please use this Ask Question box to submit your questions and comments throughout the webinar, and our presenter will answer these at the end of the presentation. We will have Q&A session at the end of the webinar. Okay, I already see first responses. Thank you very much. And before we begin, I would like to quickly go through the disclaimer. QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of a disease. And we are joined today by our speaker, Dr. Domenica Martorana, who is an R&D scientist working on gene regulation product development at QIAGEN Hilden in Germany. She studied molecular and cellular biology at the Georg-August University in Göttingen in Germany, where she received her PhD in 2019. Her PhD research focused on the transcriptional regulation of the unfolded protein response in both fungi and higher eukaryotes. Along the way, she has gathered extensive experience in gene editing and gene regulation in different international projects. Since joining QIAGEN in 2019, Dr. Martorana has been involved in developing CRISPR and functional gene regulation-related products. And now I will hand over to Domenica.
So, good morning, everyone, and thanks, Paula, for the kind introduction. I would also like to welcome all of you to this webinar. I'm glad to have the opportunity to present to you how to easily characterize CRISPR gene edits with edit speed. But before doing that, let's start with some basic information on CRISPR. CRISPR stands for Clustered Regularly Interspaced Palindromic Repeats and was firstly discovered in the late 80s and later on described as potential bacterial immune system to fight viral infections. The principle is quite easy. So an unknown phage infects a bacterial cell for the first time. The DNA of the invader is being inserted into the CRISPR array, and from that moment on, the cell gains immunity against this type of invader. Next time this type of phage is attacking the same cell or its offspring, the CRISPR machinery is activated and serves as a kind of immune system. And this system has laid the foundation for how to use CRISPR as a molecular biology tool in genome engineering. Genome engineering helps understand the role and function of a gene. So in a reverse genetic approach, for example, a gene of interest would be deleted and the effects of such a mutation analyzed. Questions that would come up are, how does the gene affect morphology or the transcriptome? Does the gene have an impact on the cell cycle or the metabolism? Do I see, for example, changes in the proteome or in signaling pathways? What's actually happening on a genome level? And is interaction, be it on a genetic or on a cellular level, impaired? And the mutation of a gene, as you can see here, can have a bunch of effects on cells. So it is essential to use a reliable system that just introduces the wanted mutations without having additional side effects and also without interfering with other cellular mechanisms. Having a closer look into techniques for genome editing in mammalian cells, there are three prominent techniques called zinc finger nucleases, TALENs, and CRISPR. And all three methods are able to specifically introduce mutations into the genome. On the left and the middle, there are zinc finger nucleases and TALENs. They use for that a split nuclease that is directed to the region of interest in the genome by proteins that specifically recognize a sequence motif within the gene of interest. And both systems are well suited for genome engineering. However, the design of the left and the right part of the constructs, which guides the split nuclease to the region of interest, is rather tricky, I would say. The CRISPR-Cas technology can also be used for gene editing, but it works a bit differently. So here we have a guide RNA that leads the endonuclease, and in this example on the right side, it's Cas9, and this Cas9 is guided to the gene of interest. And for that, a protospacer adjacent motif, short PAM, must be present in the genome, but not in the guide RNA. So once the PAM is present and the guide RNA is perfectly matching to the region of interest, the endonuclease is introducing a double-stranded breaks, and this double-stranded breaks needs to be urgently repaired by cellular repair mechanisms such as non-homologous end joining on the left and homology-directed repair on the right. When repaired by non-homologous end joining, the double-stranded break is randomly repaired by either removing or adding some base pairs. So the outcome in such an experiment would be cells carrying all different kinds of indels, and we will see that later in this presentation. When repairing via homology-directed repair, then a donor DNA is additionally needed and delivered into the cells that serves as a template on how to fix the break in the genome. And with that, the outcome goes more in a yes or no direction. So either it's edited or it's not edited. And both methods are widely used depending on the individual experimental setup. So depending on what kind of genome engineering is used, the CRISPR strategy is changing. And unfortunately, there is no one CRISPR workflow that would fit all scenarios. But what all of the approaches have in common is that they all start with the target gene. And before starting with the editing event itself, it is important to choose the corresponding editing strategy. And it really depends on the experimental setup. Sometimes a plasmid strategy might be preferred over an RNP setup, and of course, vice versa is also the case. Depending on the target gene, the choice of the guide RNA, and with that also, of course, the choice of the endonuclease is crucial. And depending on the cell type, and of course, the CRISPR method of choice, there are several possibilities on how to get the CRISPR components into the cells, such as lipofaction, electroporation, nucleofaction, and so on. So some cell types, for example, can only be transformed via electroporation, whereas other cell types do not tolerate electroporation at all. All those steps have a major impact on the analysis of editing events. So the more I optimize the 3 steps along the CRISPR workflow, the better the outcome and the less effort is needed for screening and finding the cell carrying the mutation of interest. When I did my first CRISPR experiment, and this was back in 2017, I was working on a transcription factor regulating the unfolded protein response. And I wanted to quickly generate a knockout cell line to be able to analyze the effects of this transcription factor. I wanted to be done with that in just a couple of weeks, since from literature, it did not seem to be too difficult. I wanted to delete my gene of interest in 2 different cell lines with 5 deletion approaches, including, of course, non-homologous end joining and homology-directed repair. And I used several CRISPR editing techniques and 2 different delivery strategies. I wanted to be on the safe side, just in case that one of the strategies is not as straightforward as expected. Also for cell clone isolation, I used several methods, and it took me, I think, about a month before I could start screening for the cell clone harboring the edit of interest. And back then I was quite confident that it is just a matter of days until I have my knockout cell line in hand. However, as you might imagine, the screening process took an unexpected turn. So since all the available antibodies against my transcription factors were not working, I had to screen on a DNA level. I had multiple DNA extraction methods combined with different PCR chemistries, and clone number 670 was finally the knockout clone I was looking for. And all in all, it took me around 2 months of cell cultivation and screening, and I can tell you 2 very laborious and frustrating months. And it was just later on that I realized that this was not just me with the deletion of my favorite transcription factor, but challenges along the gene editing and screening process can occur. And sometimes, no matter how much you optimize or how much optimization is involved, several hundreds of cell clones need to be cultivated and screened to obtain the cell with the edit of interest. There are different factors that can make screening for the gene edit of interest long and tedious, and some of them are listed here on this slide. So let's start with low editing efficiency. Low editing efficiency may result in a greater screening effort. However, sometimes boosting the editing efficiency might also increase unwanted off-target events, and this can negatively impact, for example, cell fitness. It can result in a reduced survival rate or also in a reduced cell growth and proliferation. And this, in turn, significantly increases the cell cultivation effort. And one thing that we should always keep in mind is that the longer we cultivate the cells, the older they are. And of course, they have more time to collect mutations, and those mutations can affect the whole downstream analysis. And last but not least, sometimes lots of cell clones, as I told you already, need to be screened. And the target gene is in most cases amplified by a Pcr. And for that, you need specific, but at the same time also very efficient primers. And this primer design sometimes requires expertise to avoid some major PCR optimization later on. And depending on the experimental setup, the gene of interest and the editing to be introduced, you can face a variety of challenges. Most editing workflows can be roughly split into 3 steps. Once the CRISPR editing has taken place, the cells need to be processed and the DNA extracted. And in most cases, the lower the editing efficiency, the higher the screening effort. Next, the target of interest is amplified via PCR. So for that specific primers and efficient primers, as well as a robust PCR chemistry are needed. And last but not least, editing needs to be somehow analyzed. And for example, via Sanger sequencing. And here again, Sanger sequencing primers are required, and the sequencing traces also need to be evaluated. In the following, I would like to guide you through the 3 steps one by one, highlighting some bottlenecks and providing some tips and tricks on how to simplify and to speed up the process. So let's start with sample preparation. There are many different methods. I know that there are many different methods available on how to extract DNA from different cells. And for CRISPR screening purposes, the easiest and fastest way to do that is to directly lyse the cells and to use the raw lysate for target amplification without additional purification steps. So one of the major differences here between the various sample preparation techniques is the amount of cells to start with. And this is very crucial, especially when dealing with high mortality after CRISPR editing or when working with single cells as shown here. So after clonal expansion, you start with just one single cell. And for example, a sample preparation method that requires 1,000 cells would involve 11 days of cultivation for cells that have a generation time of 24 hours. Don't worry, I added 1 day here, since we always need to keep the clone alive. If all cells are lysed, without the backup, there's nothing left after cell lysis, so one would need to start with the editing all over again. And at that example, it would, of course, be beneficial to choose a sample preparation method that only requires a couple of cells, reducing cell cultivation time and also resulting in younger and fitter cells. We at QIAGEN developed a liquid-based sample preparation method that enables DNA preparation from just a few cells. And as you can see here on this slide, a broad range of cell input is applicable. And we can even go below 10 cells per microliter of lysis buffer. So the sample preparation is compatible with different experimental setups. So we can go with 6 well plates, 24 well plates, 96 well plates, every kind of imaginable experimental setup. And the sample preparation takes less than 30 minutes. Even, and I was also surprised, but I experienced it myself, even when preparing 96 samples at one go. Next, now we have the lysate, and next, the target of interest needs to be amplified via PCR using the sample prepared in step 1, but without additional purification steps. So this would be the optimal scenario. And what do we need for that? So first of all, we need a PCR chemistry that is sensitive enough to deal with the raw cell lysate containing just a few cells. And with that, of course, also containing just a limited number of target copies to be amplified. So as you can see here, we combined the raw cell lysate from step 1 with QIAGEN's all-tag PCR chemistry. And we prepared the lysates with different cell numbers per microliter of lysis buffer from different cultured cells, such as EukaryoTe, HeLa, U2S cells, and so on. And we always transferred 5 microliter of lysate into the PCR reaction. In this example, you can see the amplification of EMX1 from half a cell on the left side to 50,000 cells, per reaction on the right side. And as you can see, 1 cell is already sufficient for successful amplification of EMX1. And this, of course, may vary depending on the target of interest. However, also with other target genes, few cells are already sufficient for successful target amplification. For the amplification of EMX1, we used in this experiment CRISPR-Q custom-designed Pcr primers, but this would also perfectly work with, let's say, good self-designed primers, but I will come back to the primer design topic in a minute. Next, the PCR chemistry should also be robust against inhibitors that might affect PCR performance. So when cultivating cells, different types of potential inhibitors, such as transfection, transduction, coding agents, and so on, can be carried over into the PCR. And we tested the inhibitory effects of different components, as shown in this list. And I would say I have good news for you. So our all-tag PCR chemistry is very robust towards these reagents. And this allows us to use the DNA from the raw cell lysate without any further purification prior to PCR, and also without the addition of laborious cell washing steps. And this is very important, not just because it's always good to save time, but especially when working with few cells. Being able to skip the washing step is a great benefit, since every washing step also leads to loss of cell material. And this is something that you definitely want to avoid as good as possible. And last but not least, a PCR chemistry that allows flexible lysate input is very beneficial. So usually, as I said, we would transfer 5 microliter of raw lysate in a 20 microliter PCR reaction. But what if I want to save on lysate because I do not have too much, or the lysate is too thin and I need to use more to avoid undercutting the lower limit of detection? So in that case, we would need a system that allows to use either less or more lysate in the PCR reaction. And this is exactly what we can do when using the all-tech PCR chemistry in combination with the lysate prepared with QIAGEN's new liquid-based sample preparation method. So as you can see here, the lysate volume can be increased from 5 microliter to 14 microliter in just a 20 microliter PCR reaction. And even if not shown, the same holds true with reducing the lysate volume in a PCR reaction, for example, from 5 microliter to just 1 microliter. And that, very important, without affecting, of course, the PCR performance. Now, we have the lysate and the PCR chemistry. And as I already mentioned, for a successful target amplification, the corresponding target PCR primers are needed. It all starts with the genomic DNA sequence, as you can see on the left side of this slide. And of course, the identification of the editing sites. Then 2 primers, left and right, with specific melting temperatures are placed next to the editing site. And the primers should amplify a reasonably long fragment, which means the fragment should be as short as possible and as long as needed. And this looks very simple on this slide. However, to design specific and efficient primers, some kind of design expertise is required. That was the reason why we decided to develop on GeneGlobe a CRISPR-specific design tool for human, mouse, and rat targets. And for the design on GeneGlobe, only the species, the genomic location of the gene added, and the guide RNA sequence is used, or the guide RNA sequence that was used for the CRISPR editing experiment is needed. And based on that, in silico validated CRISPR-Q custom PCR primers are fully customized, designed, and those primers would generate then a PCR product of 400 to 600 base pairs. The PCR products are later on compatible with Qsanger sequencing analysis, but also with mismatch cleavage assays such as a T7 assay. And this directly leads me to the last step in our workflow, which is the editing analysis. Once the target has been amplified via PCR, additional analyses are required to see whether the editing of interest was successful or not. And therefore, Qsanger analysis or Qsanger sequencing is suitable. For that, as I already said, Qsanger sequencing primers are needed, and the GeneGlobe primer design tool that I showed you for PCR primer design is also designing Qsanger primers so that for every PCR primer pair, there's also the corresponding, as we call it, best matched CRISPR-Q Qsanger primers. And since we always recommend to sequence a PCR product from the 5' as well as from the 3' end, both primers are designed and would result in sequence traces with more than 100 base pairs of high quality data. Those Qsanger sequence traces can also be analyzed on GeneGlobe using the CRISPR-Q Qsanger analysis tool. For that, only a Qsanger trace of control and sample, and again, the guide RNA sequence that was used for editing is needed. And based on this information, the non-edited sequence trace is aligned with the editing sequence trace. And this editing efficiency is then calculated on an allele level. In this experiment, for example, we performed a non-homologous end joining CRISPR editing and analyzed the editing efficiency in a cell pool. And on the left, you can see the indel spectrum, and approximately 20% of the alleles had either one base pair inserted or deleted of course, always compared to the non-edited wild type control. And on the right side, you can find some more information on what exactly was inserted in case of a one base pair insertion event. In this experiment, 50% of the alleles had an adenine insertion, 25% of the alleles had a guanine insertion, and in 12.5% of the cases, either a C or a T could be found at this position. And with those results in hand, it is easier, for example, to decide how many cell clones need to be screened, if the editing experiment needs to be repeated, or if it's worth considering maybe a totally different editing strategy. In a nutshell, once the CRISPR editing experiment has been performed, the screening for the cells containing the edit of interest starts. We at QIAGEN work on a characterization workflow that unifies sample preparation, target amplification, including the primer design, and editing analysis. So our new QIAprep and CRISPR kit contains everything needed for the liquid-based sample preparation from cultured cells derived from human, mouse, or rat cells. Here, the cells are washed and lysed by the addition of the provided lysis buffer. After just 15 minutes of incubation at room temperature, the lysis reaction is stopped by incubation at 80 degrees for 10 minutes. The raw lysate does not need any further purification step, as I could show you, and can either be directly used in a PCR or stored at minus 20 degrees. Everything needed for the target amplification is also provided in the QIAprep and CRISPR kit, and the cell lysate together with some target-specific primers are then used in the PCR reactions. The primers can be either self-designed or designed using the CRISPR assay design tool on GeneGlobe. And the PCR chemistry and the lysate are, of course, particularly suited together with the CRISPR-Q custom PCR assays. Besides the PCR primers, also Qsanger primers can be designed with the GeneGlobe tool. And last but not least, the Qsanger sequence traces can be analyzed using the Qsanger analysis tool, which also can be found on GeneGlobe. All in all, we developed an innovative, fast, and sensitive characterization workflow that combines a liquid-based sample preparation with target amplification, primer design, and also Qsanger analysis. And all that looks at the end like that. The small blue QIAGEN box contains everything needed for sample preparation, target amplification, and is called QIAprep and CRISPR kit. And the custom PCR primers, CRISPR-Q custom PCR assays for PCR, and CRISPR-Q Qsanger and the CRISPR-Q Qsanger primers for Qsanger sequencing are optional and not included in the QIAprep and CRISPR kit. With that, I'm at the end of the presentation, and I would like to thank all of you for participating in today's webinar. So if you have questions, please use the Q&A function, and I'm very happy to take them. So, okay, I see one question here that is, can guide RNA longer than 20 base pairs be used with Cas9, the wild type from SP, and still efficiently generate double-stranded breaks? So, yeah. With every endonuclease that can be used for CRISPR editing, there is also a perfect matched guide RNA. So if there is if you're using a Cas9 that tolerates guide RNAs longer than 20 base pairs, then it should work. There are also Cas12 enzymes that work with a bit longer guide RNAs. So it really depends on the endonuclease that is being used. Then we have another question. Do you need to purify your validation PCR products? So as I said, prior to PCR, there is no purification needed. However, for example, if you're going with Sanger sequencing, then of course there is a purification is definitely needed, because you would carry over the PCR components into the Sanger sequencing, and the Sanger sequencing would majorly be affected by the dNTPs, by the buffer, by the primers and everything. So before PCR, no purification is needed. After PCR, purification is needed. And if you decide to use the PCR fragment for T7, for example, for a T7 assay, then there's also no need to have a purification, since the chemistry that we used is mostly compatible with a T7 assay, for example. Then we have, does the CRISPR kit work on prokaryotes? So our QIAprep&M CRISPR kit is optimized for usage with human, mouse, and red cells. And so I mean, at the end, if the lysis buffer, of course, can also work with other types of sample input. And also the PCR chemistry is not just limited to higher eukaryotes or to human, mouse, and rat. However, the primer design on GeneGlobe, this would not work. So the primer design tool that we have on GeneGlobe is very limited to human, mouse, and rat cells, but the rest, the lysis buffer and the PCR chemistry from the QIAprep&M CRISPR kit could also work with other than human, mouse, and rat cells. So then here we have another question. When characterizing edits using Sanger sequencing, no cloning of PCR products is required. Yes. Actually, when I did my CRISPR experiment during my PhD, I was also cutting out the bands of a gel and cloning it in a vector and then sequencing it from a vector. I learned at QIAGEN that this is not needed if you have specific PCR products, and this is a must to have a clean, high yield PCR product. And when you have that, there is no need to go with additional cloning and extraction from gels or something like that. So the PCR product can directly be analyzed in Sanger sequencing if you have a specific PCR product. If there is additional PCR products, unspecific products that you can see on a gel or also when using the QIAxcel, then of course one would need to optimize the PCR or to go with the purification maybe from an agarose gel, just the old-fashioned way that always works. Then we have here another question. Is the QIAGEN CRISPR series kit for research use or clinical use? So the kit that we have is, as disclaimed from Paula, is for research use. Here we have another question. I'm working with long inserts, knock-ins, and have challenges to amplify them. Is your method limited to a specific insert length? This is a question that we get quite often. As you might all know, the longer the PCR fragment, the more difficult it is, of course, to amplify it. This is just how it is. However, our all-tech PCR chemistry in combination with the lysis buffer that we have is able also to amplify longer fragments such as 5 kb, and the limitation is with 9 kb. So our all-tech master mix together with the lysis buffer in the QIAprep&M CRISPR kit is able to amplify even 9 kb long PCR fragments. However, of course, as you can imagine, there is a bit more optimization required. For example, one PCR cycling, of course, cannot be used for the amplification of 500 base pairs and 9,000 base pairs at the same time. So this is, of course, not the case, but we have in our handbooks, in the all-tech handbook and also in our CRISPR handbook, some guidance on how to get also long PCR fragments amplified without major troubles. Then we have this one. Let me see if we have more questions. Is it compatible with other PCR reagents such as X-Tech from Takara? So in our QIAprep&M CRISPR kit, there is the lysis buffer and the all-tech PCR chemistry together. So with the lysis, we have an easy and reliable way on how to extract DNA, and this DNA is of course also compatible with other chemistries. However, we optimized our chemistries to perfectly work together. So this is what we worked on. So you will get best results when combining the lysis buffer together with the all-tech chemistry, which is very robust and is even able to amplify long fragments. And this combination works best. Okay, I see another. Here we have another one. Can I use the QIAprep&M CRISPR and the CRISPR-Q primers for the characterization of gene edits that were introduced with talents? Yes. So the QIAprep&M CRISPR kit has CRISPR in its name, but however, as you've seen in my very first, in my second introduction slide, starting from the editing, the characterization is the same. The only thing that you need to keep in mind here is, for example, when using our primer design tool, we need the guide RNA sequence. And in that case, you would just take the sequence around the cut site that was introduced with talents or also zinc finger nucleases. So the QIAprep&M CRISPR kit works also for talents editing and also for zinc finger nucleases. However, when using our primer design tool, then just use a region around the cutting site and use this as a guide RNA. So take 20 to 25 base pairs and just go for it. It will work. So how many types of samples have you tried using the lysis buffer? What are they? So we optimized our QIAprep&M CRISPR kit for cultured human, mouse, and rat cells, adherent, semi-adherent, I forgot, semi-adherent and suspension cells, sorry. And we worked with clumping cells, with slow dividers, with very fast dividers, and the kit is compatible with all kinds of cultured cells derived from human, mouse, or rat. Then we have another question here. Is QIAGEN's CRISPR characterization workflow also applicable to tricky targets? I am working on GC-rich target that form stable secondary structures. Yes. So one component that we also added to the QIAprep and mCRISPR kit is a Q solution. And this Q solution is sometimes helping with tricky targets that have high GC content or form stable secondary structures. So this is one option, how to deal and how to be able to amplify tricky targets. And there is also alternative PCR cyclings that you could use, for example, increasing the denaturation time of your PCR cycling. And we also have guidance for that in our handbook. So check it out. There's definitely some options that we offer to deal with tricky targets. Exactly. So sorry, I had this question. I just forgot to click on that. Here we have a good one here. So can the CRISPR kits be used for cells edited with several guide RNAs or a guide RNA pool? Yes and no. So the QIAprep and mCRISPR kit works with every kind of CRISPR editing. So it doesn't matter if I worked with 2 guide RNAs, the lysis and the PCR is not affected afterwards. However, when choosing to use the primer design tool on GeneGlobe, then we have some specifications for that, since the design algorithm works in a way that the gene editing site is identified and the primers are designed left and right of this editing site. So if I use 2 guide RNAs, I would lose a piece of DNA. And there, of course, chances are there that the primers are located in the piece of DNA that I lost. So when using multiple guide RNAs, then QIAprep and mCRISPR kit is fine. And the primer, the Qsanger sequencing primers and the PCR primers here, I would say to take care and see if it works, or in case of doubt, just to contact us and ask if this would work or not. Yeah, here. Is there anything to consider when processing cultured cells on coated cultivation dishes? Are additional purification steps required to eliminate or reduce coating reagents such as poly-L-lysine? That's a very good question. And I was also, at the beginning, I was a bit concerned to just, for example, have a coated 6-well cells on it and just to scrape it off and have this high concentrated coating agent in my PCR reaction, but it perfectly works. And you saw the whole list of reagents that we tested. So if you have coated cultivation dishes, you will carry over the coating agent into the PCR, but the Alltag PCR chemistry is robust enough to deal with that. So here, no additional purification steps are needed, which I really like, because if you have cells that are not nicely sticky, then just and you want to use a coating agent, it's not a problem for the QIAprep and mCRISPR kit afterwards. Okay. We had this one. My computer is a bit slow, sorry for that. We have here one question, not sure if I got it correctly, but let's give it a try. So how do you solve problems with multiple alleles which are not always all edited? Dirty Qsanger. So we have the Qsanger analysis tool on GeneGlobe. And this analysis tool is able to also analyze Sanger sequencing coming from a pool. So in this pool, as you can imagine, you will not have just a clean trace, but there will be several traces depending on the editing that has taken place in the cell. And here the algorithm is identifying the cut site, so the site from where on the sequence starts to get ugly, let's call it ugly, and it aligns the part before of the upstream of the editing, and then it starts calculating on all kind of different events that happened after the cut. So the Sanger analysis tool is able to use this decomposition strategy to separate different Sanger traces and to get a editing efficiency result for that. I hope this answers your question. Do you have any recommendations for working with single cell clones when using cells that have trouble proliferating when not in clumps, such as in iPSCs? Been there with clumping cells. Is it's not very easy, but at the end, this is how the cells are behaving. And there is no right question, no right answer for this question, since it really depends on the cell type. So for the lysis, it doesn't matter if it's in clumps or not, because when you put the lysis buffer on the cells, you just also pipe it up and down, and this mechanical pipetting would efficiently lyse the cells within the clump. So the problem is more on the cultivation side, but I think this is a topic for a completely other webinar, because it's so diverse and there's so many things that one could do. It really depends on the cell line. But for the lysis, no worry. If you have clumps, doesn't matter for the downstream analysis along the workflow. And also when you need to estimate the cell number, you don't need to be like precise to one single cell in the clump. It's also just sufficient to start adding the lysis buffer, and then at the beginning it will be viscous, and then you just add more lysis buffer, and when you see that the viscosity is going down, then you're perfectly fine, and this is the lysate that you want to work with and do the PCR on. So okay, would you ever select such heterozygous clones for downstream work? Yeah, this is maybe a more personal questions, let's say it like that. I'm coming from fungal genetics, and as a fungal geneticist, we want to have it very clean, and it's only homozygous edits are considered real edits. However, I know that in higher eukaryotes, this is very, very difficult, and it really depends on what you want to do. Of course, if, for example, during my PhD, I wanted to see what kind of effects I get when deleting my transcription factor. Of course, having it in a heterozygous state might have kind of a knockdown, and I have reduced levels of the transcription factor. And I'm pretty sure that also then you would see some differences in the transcriptome or in the phenotype. However, when you really need a knockout, then I think, the heterozygous clones, they still produce the gene of interest and the protein of interest. So it really depends. For example, if you have a knock-in and you do a GFP tag, then of course you can screen and you do morphological analysis at the microscope, then just screen for the greens and it's fine. However, if you have a mixture of homozygous and heterozygous clones, then there's also always the danger that the heterozygous clones have an advantage in growing, and that's after a couple of passages, then you will you will lose your homozygous clones, and you will end up just with heterozygous clones. So it really depends. There is no right or wrong answer to this question, I would say. I'm working with cell pools, and I'm wondering what I need to consider when I screen my editing with your kit. Great. So I focus in the presentation a bit more on what to do if I just have little material or just less cells. But our QIAprep and AMP Crispr solution is, of course, also applicable to Crispr experiments where lots of cells are present, such as when working with cell pools. There is nothing much to consider. It's just a lysis step. So if you have 1 million of cells, then it's a couple of more microliter of lysis buffer are needed. And we would suggest to go with 10 to 3,000 cells per microliter of lysis buffer. Of course, you could have also more cells per microliter of lysis buffer, but at some point, the lysate, as I already said, gets very viscous and bubbly. And then also it's more difficult to transfer the lysate into the PCR reaction and so on. But we also have a protocol in our handbook on how to just simply dilute the amount of cells and to have a reasonable cell number per microliter of lysis buffer, and this is absolutely no problem. So there's not much that needs to be considered. And if you feel like that it's still too viscous, just add more lysis buffer, as simple as that. Okay, I think. Do you see any more questions, Paula?
No, and I think we are also running out of time. I would like to quickly mention to our audience that we will send you in the next days email with webinar recording. And with that, I want to thank you all for attending our webinar. And I also want to thank you, Domenica, for joining us today. I will close now the session, and I wish you all a great rest of the day. Thank you again.
See you, bye.
Bye-bye.